3:30, so I guess we'll go ahead and get started. I'm happy to introduce Dr. Branwen Williams from Claremont Colleges, and she's actually affiliated with 3 different colleges. It's a big hippie consortium of colleges out in California. I met Branwen in grad school. We did our PhDs together, and then like any sensible person, she tried to get as far away from me as possible, so I came here to do a postdoc, and she went to Canada. But that wasn't far enough, so then she moved out to California for a faculty job. With that, I give you Dr. Williams.
Okay. Thanks, Ryan. Thank you for the introduction. And thank you all for coming today. I've had a great day touring around all the different institutions that are here. I'm going to talk to you today about some work that I've been doing for quite a few years now, and that's developing and using these coralline algae as archives of changes in climate. Now, I'm probably not telling you, I'm assuming I'm not telling you anything new and telling you that our temperatures are going up. In fact, 2015 summer was the hottest on record. With the El Nino event in the Eastern Pacific, this is on track to be a very, very warm year. So we know our climate's changing. We know people are causing that change. But when we look at changes in temperature, one thing we also know is that it is not equal throughout our globe. And these are projected temperatures for the end of this century, so it's not so far away now, about 85 years for the 2100, under 2 different greenhouse gas emission scenarios. This is our 2013 IPCC report, which I'm sure most of us have spent time looking over, for lower emissions up top, and then higher emissions down at the bottom. And we can see that in the Arctic, the northern hemisphere, high latitudes are projected to warm the most, quite significantly more than the global average. There's large spatial variability in that projected warming. And this is a reflection of, in part, of the ice albedo feedback in the Arctic. The melting sea ice is going from a nice white reflective surface, which sends a lot of the radiation back up into space. As it melts, it turns into darker ocean water or brown land, which absorbs more of that radiation, which means it heats up more, which means it melts more ice. And that positive feedback is going to contribute to some of this Arctic amplification. And these temperatures are projected under the high emission scenario of about 10 degrees warmer than they have been, which is a pretty big difference. So we really want to understand the climate mechanisms at these higher latitudes that cause climate change so that we can kind of improve these projections. And to do this, we need to have data. The climate models are built on our understanding of the physical mechanisms of climate that can be understood from using time series data. And then also we can validate, verify the model output using time series data. And that's the field that I'm in. I use climate archives to create time series data so that we can understand our climate. This is a figure showing the number of years of the ICOAS data set of temperature observations. The red goes back to about 110, 120 years of seawater temperature measurements in the oceans. The yellows and greens are about 60, 70 years. And then the blues are 10, 20, 30 years. And there's a couple of observations down in the Southern Ocean. There's no data, so we don't really know what's going on down in the Southern Ocean. I know there's some really great work here being done in the Southern Hemisphere. I've so far focused more on the Northern Hemisphere. We're going to talk today about the Aleutian Archipelago, where we have data going back to about 1960, so about 50 years of observations. And then if you look into other parts of the Arctic, the Canadian Arctic, again, it's another area where we have very little data. The reason why, or one of the reasons why I focus on the Bering Sea, it's a very highly productive system. Deep water, because of the shape of the seafloor, comes up to the surface, and that deep water brings a lot of nutrients which can support a high amount of primary productivity. So it's a very, very nutrient-rich ocean, which means it can support ecosystems, and a lot of the US fisheries are based in the Bering Sea. So ecologically and economically, it's a very important region. It's the oceanic gateway between the North Pacific Ocean and the Arctic Ocean. So any North Pacific waters are going to flow through the Aleutian Archipelago into the Bering Sea and then into the Arctic Ocean. Our passes through the Aleutian Archipelago, so water coming from the Gulf of Alaska can flow along and then slip through these different passes into the eastern Bering Sea shelf in here before going up into the Arctic. So oceanographically, it's a very important region. In terms of the climate, it is at the intersection of the locations that can feel the impact of different climate patterns. For example, the Pacific Decadal Oscillation, it's at the northern extent of where the PDO influence is, and then at the southern extent of where kind of the Arctic Oscillation. So it's kind of that intersection of the North Pacific Ocean climate patterns and then the Arctic Ocean climate patterns. So climatically, it's a really interesting location. And it also has influence by or can help alter storm tracks, which come down and can influence the US West Coast. And coming from California right now, we're really interested in what's happening with those storm tracks. So to really understand what is going on in the Bering Sea, to understand past changes so we can understand climate mechanisms, and then improve our understanding of how it's going to change going forward, we really need time series data. We need temperature that extends prior to the 1950s. If we're seeing a PDO influence, 1950s is really only 1-2 PDO shifts. If we're seeing Arctic oscillations, these patterns are recurring patterns that are multi-decadal, so we need longer time series to understand them. And we also need to understand other variability in the ecosystem, not just temperature. And one thing to point out is that this area is has high spatial heterogeneity. You get a lot of fast-moving currents that flow along here, as I mentioned, through the different passes in the Aleutian Archipelago, depending on the depth of the passes. But you can see the very, very high spatial variability of the Alaskan current, and then on the north side of the island, where this kind of sees Bering Sea water, and the south side of the archipelago sees more North Pacific Alaskan water. And just for reference, this is a 2x2 grid cell. So when we're talking about our gridded satellite data that does extend back to 1950 or the shipboard observations, they're generally at a resolution of 2x2, or sometimes it's 1x1. And that's going to average out a significant amount of this heterogeneity in the ocean system. So if we think about the ways that we can get information from the past, where people have not been standing out there with a thermometer, as people haven't been doing in the Aleutians, we need to turn to our proxy archives. And historically, many of these archives have been terrestrial when we're talking about these timescales of decades, centuries, maybe a couple of centuries. And this is the Mann paper from 2009 in Science, his composite that generated the hockey stick curve. And most of these archives are terrestrial. There's some sediments and there are some corals, but a very strong bias towards terrestrial, not really capturing what's happening in the ocean. More recently, there's been some fantastic work, this was published this year in 2015, where they are starting to take together the oceanic archives to paint a picture of how the oceans have been changing. And this is part of the PAGES 2K Ocean Consortium efforts. But what you notice here is that I've shown a figure with the sampling resolution, and only the red dots have more than 8 SST points per 200-year bin. So these are not, in the world that I work in, not very high resolution at all. Really, to understand how the decadal patterns are changing, we need an annual archive. And so that's what we're trying to work toward, is an annual archive at the higher latitudes. And to do this, we're turning towards 2 types of organisms, the bivalves. There's a really great bivalve community who's been doing a lot of work, and that's marked by the blue circles on here, some of the locations. We have our gooey ducks and our Arctica Icelandica, and these are showing records that are longer than 100 years old. And then the red dots are showing the records that we have so far from the coralline algae. This is an example of a rhodolith, and the rhodoliths are algae that are not adhered to the sea floor. Instead, they can roll around in shallow waters, and then they'll eventually be deposited, and then they can build up in the sediments over time. But each individual is free rolling. The algae that I've been focusing on is this genus of algae called Clathromorphum. There's 2 sister species. This is Clathromorphum compactum and Clathromorphum nereostratum, and this is the one that I'm going to focus on today. As you can see in this picture, and it was on my title slide, this algae can grow big, can grow quite large, boulder-like from the Aleutian Archipelago. So what we're working on is trying to develop these algae, develop the bivalves, so that when you come back to this figure, you can also see that although there's really great global representation, that there's still nothing from this part of the world. So trying to fill in that going forward. So this talk, for the rest of it, is divided up into 4 parts. I'm first going to tell you about Claremont, this algae as a proxy archive, why it is proving to be really useful in telling us about past climates. And then I'm going to give you parts 2 and 3 as 2 examples where we've used it to better understand the Aleutian Archipelago system. The first one is using growth rates, and then the second one is magnesium calcium. And then I'm really going to quickly talk to you about, for the part 4 of this talk, of where we're going. What is kind of what's on top, because we have some exciting work that's in progress. So for part 1, Claremont as a proxy archive, this is what the algae looks like underwater. We're pretty fortunate in that sample collection is relatively easy. It's remote location, so getting there is the battle. Sometimes we're having to, colleagues have gone diving under ice to get it. But it adheres to the seafloor, it stays in one location, and will grow there its entire life. We use highly technical devices to sample it. We use kind of a chisel and a rock hammer underwater to kind of pop it off the rock, and then we can bring it up to the surface. Living, they're red underwater, so they're quite easy to visualize. And then when we bring it up to the surface, we can just dry it, so pretty simple storage techniques. And then it loses some of its color once it's dyed, so it turns into a little bit of a green color. But we know if there's any red tissue that it was live collected. And then what we do is similar to a lot of other proxy organisms. We section it, we polish it, and then we image it to look under it under high magnification. And this is an example of a cross-section of one of the algae. So we would have crossed it under this way and then polished one of the sections. This is the oldest part of the plant that would have adhered to the sea floor, and then it grew up this way, and this would have been the living surface. One other thing that makes these plants easier to collect than a lot of other organisms is they photosynthesize, so they're within usually shallow water, about 10 to 20 meters deep. So within scuba diving range, as long as you can get to the locations where they're found. So I'm going to zoom in on kind of the outer surface of Claremont to look a little bit about how it calcifies. We make a lot of comparisons between the algae and corals. They both have a calcium carbonate skeleton, but remember that Claremont is a plant, and so it's a useful comparison, but it's very limited when you try and compare your plants and your animals. So this is SEM of the top of one of the algae specimens. This is your site of calcification, this little gap in here, and the algae is going to grow high magnesium calcite crystals. We'll calcify them both going up and then down. And the part going down, this is the main body of the plant that's going to build up over time. And what's really neat about these algae is it also forms this part coming up called the epithelium. And this epithelium keeps getting grazed off or slothed off by sea urchins or chitons when they're growing on the sea floor. But usually that grazing is just limited to this region of the plant, meaning that the bulk of the plant is preserved, undisturbed, with minimal diagenesis. In some cases, the grazing does extend down into the bulk of the plant, and you can see little scars where the grazing is extended down, but we can usually identify those pretty easily. So we're able to do visual examination of our specimens to make sure we avoid grazing. With the cell structure, they form two kinds of cells. In the summer, the plant's going to grow faster, and it's going to grow longer cells. And then in the winter, it grows slower and it grows fatter cells. And the combination of the difference between these two types of cells, that means when you look at a SEM or even just high magnification of the polyged image, you can actually see growth lines. And you might kind of have to squint your eyes a little bit to see them. But there's a growth line across here, a growth line across there, a growth line across there. And what that lets us do is assign time into the algae skeleton, because those growth lines are formed every year. When the growth slows down in the winter, you're going to have the mark. And so if you know when the specimen was collected, you can assign time, years to the skeleton, and you can count back into time. They also have secondary ways of developing the chronology, and these are the conceptacles, and they are holes in the skeleton that formally house the reproductive organs of the plant. And they are initiated, the formation of the conceptacle is initiated at the same time every year in the fall, so that you know a row of conceptacles also equals 1 year of growth. Now, the conceptacles are formed from diagenesis that the plant does to kind of carve out the hole, but the diagenesis is limited to within 1 or 2 microns of the edge of the conceptacle. So the skeleton between the conceptacles is pristine, and then if you want to analyze geochemistry, you want to stay away from the conceptacles. So what we know with these algae is that they grow in sunlit waters or water with sufficient sunlight for photosynthesis, about 10 to 20 meters deep in the higher latitudes, and that they form a skeleton that's preserved over time that has annual growth increments in it that we can use for developing a chronology. The other thing that's really cool about them is they can grow for a pretty long time. This is a specimen from the Aleutians, and I'll be showing some data from this specimen in a little while. But what we had is about 200, 150, 200 years of continuous growth. There was a discontinuity at the bottom of the specimen, and then uranium thorium showed us that the dates at the very bottom was about 850 years. So we have a plant here that was alive 850 years ago. In the sister species from the North Atlantic up towards the Canadian Arctic, we actually have another live collected specimen with 650 years of continuous growth. So these are long-lived plants. We think that with more field work, obviously field work up in the Aleutians, the Arctic is difficult. So with more effort, we could probably find specimens maybe reaching a millennium old with continuous growth. So the final thing that makes Plathromorphum a really useful archive is, as we've talked about, its location, where it's found, the growth in the skeleton. It can live for a long time. But we've also found that it records changes in the environment in the skeleton of the organism. So that's what you really need to have a useful proxy archive. And this is just an example of some of the things that part of the community that I work with is that we've been able to kind of extract from our archive to use to inform changes in the environment. We've used carbon isotopes to track changes in upwelling. We've used oxygen isotopes to track temperature and the seawater isotopic composition. And we're actually working right now to see if we can extract out the different components of temperature and seawater isotopic composition on the oxygen isotopes. If we can think that the primary driver of the oxygen isotopic composition of seawater is precipitation, then we might be able to extract out a salinity and a temperature proxy from the oxygen isotope. So that's something we're working on right now. We've used barium calcium to track the freshening of seawater. Barium calcium is a bit tricky because the barium concentrations is very much a reflection of the local oceanographic regime. So a lot of these data all need to be interpreted in the context of your field site. We've used boron isotopes to track seawater pH. And we have experiments right now. There are some samples up at Justin Reese's lab at Northeastern University, and he's culturing specimens under different PCO2 conditions. So we can develop a quantitative calibration of the boron seawater pH proxy. And then we've also are looking at whether PCO2 drives changes in skeletal density. Previous study from the specimens that I'm going to present on today found it actually tracked changes in climate and related to kind of climate patterns. But we're trying to refine that proxy. So for part number 2, I want to go into one example of how we've used the algae to tell us about changes in the Aleutian Archipelago. And this is a photo of the Plathromorphum from the Aleutian Archipelago. You can see the red color. There's some sea urchins, probably some macroalgae. And the water is not very clear. There's a lot of particulates in the water. So the first thing to be able to interpret growth rates is you need to understand what drives growth rates. And for a plant in the water, it's going to be a combination of light and a combination of temperature. In the Aleutians, the temperature range, it's about 3 to 8 degrees Celsius, the seasonal variability. And that's actually not that large relative to the other places the algae grows. It's only about a 5-degree difference. And the small change in temperature, and I know for people in the tropics, that's a huge change in temperature, or maybe people down in the southern hemisphere. But for other places where this algae grows, the 5 degrees is not a big temperature swing. And we find that for this site, for the Aleutians, there is not a big impact of temperature on growth rates. So that means the big variable that's trying to drive growth is light. And there's two factors that primarily will drive light reaching the seafloor. And again, this is light that's going to reach the seafloor where the algae are growing. And the local factor is going to be the amount of macroalgae, the amount of kelp. Aleutians are a ecosystem that has fluctuations in the amount of kelp, sea urchins, and sea otters. When you have more kelp, it's going to be shadier on the seafloor, and the algae is going to grow less. When you have less kelp, you're going to have more light reaching the sea floor, and the algae is going to grow faster. So we know that on a local level, a site-specific level, the amount of algae, the amount of macroalgae that are the kelp that's going to be shading the sea floor can drive growth. But if we want to know what's happening in the Aleutian Archipelago, we want to think about regional factors that might also change growth. And this is going to be related to the Aleutian low pressure system. And hanging out right over the Aleutian Islands is your low pressure system here. Just over the North Pacific, reaching up from kind of the temperate latitudes up into the Arctic. And here's kind of a better visualization of that, showing sea level pressure composite mean for 1988, 1999, showing an example of a weak Aleutian low pressure. And like a lot of our climate patterns, it can change over time. So this is an example of a weak one. The low pressure system is restricted to right over the center of the Aleutians. What happens during the weak Aleutian low pressure system is that the winds are weak and there's less clouds. If there's less clouds, you're going to have more sunlight reaching the ocean and more sunlight reaching the seafloor. So one thing about the Aleutian pressure system is it influences how many clouds are present. It also influences winds, and when you have a weak system, you're going to have less winds. What do winds do in the mixed layer? They help mix up water mixing, and that can bring nutrients up into the surface waters. When you bring nutrients up, you're going to change the amount of plankton. So here, when you have less winds, you're going to have less water mixing, less nutrients. The nutrients will be deeper in the water. The plankton will be deeper in the water, and there's going to be more light. The water is going to be less cloudy, and you can have more growth of the algae. Conversely to that, when you have your strong Aleutian low pressure index, and here the low pressure system is much more extensive, covering almost all of the Aleutian archipelago, extending further south, you're going to have stronger winds. You're going to have more clouds, and again, when you have more clouds, it's going to stop some of the sunlight from coming down to even reaching the surface of the ocean, so it's going to make the ocean darker. And then you're also going to increase your winds, you're going to increase your mixing, increase your nutrients, and then you're going to stimulate phytoplankton in the surface waters, which will also make your water cloudier. There's going to be more particles in it. And so this cartoon on the right is showing you where the distribution is of the phytoplankton. During a strong Aleutian low, it's going to be higher in the water column because nutrients can come higher up. During a weak Aleutian low, the phytoplankton are going to be lower, and the coralline algae that are hanging around 10 meters depth will see a large difference in the amount of light that are reaching them.